Gene Therapy, The Impossible Dream. Lexile 1050L. Publication Odyssey, February 98. Author Faith Hickman Bryonick. By his own admission, W. French Anderson was a weird little boy. He could read, write, and do arithmetic before kindergarten. He was chomping through college textbooks by the time he turned 8. Because he stuttered, his classmates teased him. But he didn't care. His mind was filled with dreams of science and medicine. When he was grown up and a college student at Harvard, Anderson's boyish impulses often got him in trouble. One day he was listening to a visiting scientist talk about sickle cell anemia. In that inherited blood disease, a recessive gene causes a chronic, usually fatal, anemia marked by crescent-shaped blood cells. His thoughts racing, Anderson blurted out the inspiration that popped into his head. "You could actually change the genes and correct sickle cell anemia," he exclaimed. "What a stupid thing to say," the lecturer retorted. "This is a serious scientific session." Humiliated, Anderson tried to slink out of the meeting, but one of his professors approached him. "Interesting idea," the chemist said, and walked away. That was all the encouragement Anderson needed. Then and there, he set himself a single-minded goal: to cure disease by replacing bad genes with good ones. In sickle cell anemia, a recessive gene causes a chronic, usually fatal anemia marked by crescent-shaped red blood cells. Today, Anderson directs gene therapy research at the University of Southern California. He's one of thousands of scientists working all over the world to turn a seemingly impossible dream into reality. To understand gene therapy, we must look not only at scientists like Anderson, but also at the patients they treat. In 1990, Anderson was the first to try gene therapy on a human being. His patient was 4-year-old Ashanti De Silva. De Silva was sick because she lacked the gene that controls the production of enzyme essential to the immune system. Anderson and his team took some white cells from her blood, exposed them to viruses carrying the normal gene, and then returned them to her circulation. The experiment was successful. Some of her blood cells began making enzymes she had been missing. The altered cells died off, however, so Ashanti needs continuing treatment. But today she leads a relatively normal life. Since then, hundreds of others have volunteered for experimental gene therapy sessions. Alyssa Mushin and Lindsay Karen have Canavan's disease, a defect of the nervous system that usually kills its victims before they are 10 years old. Scientists aren't sure what causes the spongy degeneration of the brain and spinal cord, but they know that children with Canavan's are low on an enzyme called aspartame. That enzyme breaks down another compound called NNA. The pieces derived from NNA are used in building myelin, the productive sheath that covers nerves. If NAA isn't broken down, myelin isn't produced. That means nerves can't carry messages from the brain and spinal cord to the muscles and bones to produce movement. Last March, Matthew During, director of the Gene Therapy and Neurogenics Laboratory at Yale University, injected the DNA that codes for aspartame production into the girls' brains. "We have indirect evidence," During says, "of gene transfer with normal levels of NAA in several brain regions in both children. That's definitely an exciting step, but only a first step." During cautions, it's too soon to think that the gene therapy can cure Canavan's. Arnold Motulsky, a geneticist at the University of Washington, says gene therapy makes good sense in terms of the theory, putting in a good gene for a bad one, but the problem is to make it realistic. Although some trials are promising, scientists are still looking for better ways to get genes into cells and get them to work once they're inside. The body's natural defenses against invaders are a problem. The immune system can reject a transplanted gene just as it rejects a transplanted heart or liver. Anderson's first approach used a virus to carry the gene into the cell. Viruses are natural choices because they are engineered by nature to infect cells and inject genetic material, DNA or RNA, into the nucleus. But they are not always ideal gene carriers. They're too small to carry large genes. They don't always seek out the right cells. Sometimes they insert themselves in the wrong part of the cell's DNA. Although altered in the laboratory to be safe, some viruses have triggered inflammation in a few patients. Last year, Dr. Chris Higgins and his colleagues at Oxford University in England tried a different approach with people who have cystic fibrosis. This disease causes the respiratory system to produce a thick, sticky mucus that clogs air passages and interferes with breathing. Higgins used spherical fatty molecules called liposomes to ferry normal mucus producing genes into the nasal passages of his patients. He then tested to see if his volunteers were producing normal mucus proteins. They were. We can deliver the DNA, its functions, it makes the protein works, Higgins says, but that doesn't mean he can cure cystic fibrosis. Cells in the nose and lungs divide and replace themselves every 3 weeks, losing the transplanted gene. So the treatment is not truly gene therapy, but merely a temporary gene transplant. Still, it's a start. Another solution to the transfer problem may be the artificial chromosome invented by researchers at Case Western Reserve University in Cleveland and Athersys, Inc. In the living cell, chromosomes in the nucleus carry genes, much like beads on a string. The scientists devised a way to make such a string of beads in the laboratory, building into a synthetic chromosome the normal bead of a gene that might take over for a faulty one inside the gene. Another approach uses liposomes to carry a short chain of DNA and RNA into the cell. For reasons not totally understood, the chain binds to a defective gene and acts as a red flag, summoning the cell's own repair machinery into action. In one such procedure, a team at Thomas Jefferson University in Philadelphia took blood from a patient with sickle cell disease, that same disorder of the blood oxygen-carrying molecule hemoglobin that so intrigued French Anderson when he was a student. They inserted the short DNA chain and waited to see what happened. They found that more than 1 in 5 of the treated cells was making the normal healthy form of hemoglobin instead of the sickle cell form. That might be enough to lessen symptoms, although a truly permanent cure would require a change in the genes of the stem cells in a bone marrow that produce red blood cells. Dr. Ronald Warwick of the University of Cincinnati is experimenting with yet another technique. We're trying to insert a suicide gene into tumor cells and then give the patient an otherwise harmless drug that the gene turns into a toxin, he says, fighting a deadly, fast-acting form of brain tumor called glioblastoma. Warwick uses a virus to carry a gene into the tumor cells. The gene causes the tumor cells to make a protein called TK. 2 weeks later, the patient gets an injection of a drug that turns TK into a poison that kills the cancer cells. Healthy cells aren't making TK survive unaffected, or that's the hope. It may be a long time before French Anderson's dreams become reality. So far, there has been no successful human gene therapy, points out Canadian geneticist Ronald Davidson. Evidence of gene action has been short-term or incomplete, he notes. The goal of a permanent change in genes has yet to be realized. It's quite possible that none of the current approaches to gene therapy will succeed, he says. Still, he believes that now and ultimately successful techniques will eventually be found. W. French Anderson is less cautious in his predictions. 20 years from now, gene therapy will have revolutionized the practice of medicine. Virtually every disease will have gene therapy as one of its treatments, he says. Workers in biotechnology industry are heading in that direction, and kids like Ashanti, Alyssa, and Lindsay are helping make French Anderson's impossible dreams come true.